Hello, welcome back to Scholars Forum. I still remain Pascal Dubai from the Department of Medical Surgical Nursing. In our previous video, we discussed extensively on the history of Mendel work. Then I went further to explain the Mendel first law of inheritance, where we talk about the law of segregation of gene. That is where alleles separate during gamete formation, that is during mitosis, whereby allele they separate in order to avoid the inheritance of double allele leading to abnormality. Now in today's video we'll continue from where we stopped. We'll be looking at the Mendel's second law of inheritance, which is called the law of segregation of gene. This law states that genes for different traits, they are inherited independently of each other during gamete formation. That is, genes for different traits, they are inherited independently from each other during gamete formation. That is, this means that the inheritance of one trait does not affect the inheritance of another trait, especially when the gene they are located on different chromosomes. That is what this law is talking about. The inheritance of one trait does not affect the inheritance of another trait, especially when the gene they are located on different chromosomes. So therefore, in exchange, the law of independent assortment of gene, this law states that genes for different traits are passed to offspring independently. They are passed to offspring independently from one another. Independently of one another. That is, genes for different traits are passed to offspring independently of one another. That is, the inheritance of one trait does not affect the inheritance of another. Yes, let me use this example to explain what I'm saying here. Is that, for instance, the inheritance of the shape of a seed does not affect the inheritance of the color. Of a seed. That is, both of them, they are being inherited independently of each other. That is, one trait does not affect the inheritance of another trait. Yes, that is, the inheritance of one trait does not affect the inheritance of another trait. Let's move to the example in your slide to explain what this law is talking about. The independent assortment of gene. That is, the inheritance of one character does not affect the inheritance of another character, which means that genes, they are inherited independently of each other. From our outline here, they use an example of pea plant. Yes, they use an example of pea plant. From this outline, you will get to find that we have two parents. One of the parent is with capital R and a capital Y, that is round and yellow, and the other parent is a small r and a small y, which is a wrinkle and the green. Wrinkle and green. Let me wipe my board. Wrinkle and green. Now remember this, that the R, the capital R, it stands for round, right? And is dominant. It's a dominant allele. Then the small r, it stands for wrinkled, and it is the recessive allele. Then the capital Y, which stands for yellow and is dominant, the dominant allele. Then the small y, which stand for green and is recessive. We are together to this point, right? Now the next thing that will happen is the formation of gametes. That is, each parent will produce gamete. Are we together? The next thing that will happen here is the formation of gamete, and each parent will produce gamete, and each gamete must carry one allele. Each gamete must carry one allele, since we are dealing with the shape and color of a seed. And each gamete, each parent will first of all produce gamete, and each gamete must carry one allele for shape, one allele for shape. That is for the shapes, for the seed shape, and one allele. That means segregation of gene occur here, right? One allele for color, for seed color. That is the next thing before crossing. Definitely there ought to be formation of gametes, right? Each parent will produce gamete, and each gamete must carry one allele of seed, one allele of the seed shape, and one allele of the seed color. That is, there ought to be segregation of gene. We are together to this point. Alright, so therefore, for the parent with a capital R, that is round and yellow, after segregation of gene occur, the parent, it will give a R and a Y. Yes, it will give a R and a Y. Look at it, after segregation of gene occur. For instance, you know RR, they are all dominant, right? So therefore, after segregation of gene occur, if you remember in the previous video, while explaining segregation of gene, I said that the law of segregation of genes states that two allele for a trait separate during gamete formation, so that each gamete carry only one allele. The reason being that when fertilization occur, the offspring receive one allele from the father and one allele from the mother. And I said the reason being that if they inherit 2, it will produce 4, which is a chromosomal abnormality. Are we together? That is why the segregation of gene ought to happen. Are we together? That is it important to avoid chromosomal abnormality. So therefore, the parents with RR, that is capital R and a capital Y, will have only R and Y after segregation of gene occur. Are we together? That is, for R, since only R is available, you will have only R. Then for the Y, since Y is available, you have only Y. If you check after crossing, you have RY. You understand? They are all the same, RY. Look at it, RY. We are together, right? RY. Then, so in essence, all the gametes will be for the parents with RRYY. It will be RY. That is during gamete formation. Then for the parents with a small r and a small y, in gamete formation, since we have only R, right, that I'm doing segregation of gene, the gene that will go to, that is the gene that the offspring will inherit, it will be R, a small r and a small y, right? That is, after segregation of gene happens, you have something like this, a small r and a small y, to avoid duplication of allele. Because if the segregation of gene did not occur, I said that it will lead to abnormality. Are we together? That is why segregation of gene ought to happen before fertilization. Are we together? That is, for the parents with a small letter R, it will give a small r and a small y. So the next thing that will happen here is, after the gamete formation, it will be fertilization, right? Yes, it will be fertilization. That is fertilization of the F1 generation. That is the fusion of the F1 generation. That will be the next thing that will happen. Fertilization of the F1 generation. Now these two gametes, they will combine. That is the capital R and the capital Y will combine with the small r and the small y, and all the generation, that is, and they will give the offspring. Their offspring will be something like this. Are we together? Yes. That is, all their generation will be RR, right? Capital Y, small y, something like this. That is, all the offspring will be round and yellow. All the offspring will be round and yellow. That is the F1 generation. All the offspring will be round and yellow. And but the round, they are not homozygous. They are now heterozygous, right? Are we together? I said that the first thing that will happen is formation of gamete. Each parent will produce gamete, and each gamete must carry one allele for the shape of the seed, since we are dealing with the shape of the seed and color, and one allele for color. So for parents with double R, a double capital R, that is with round and yellow, after segregation of gene, it will have a round and a yellow altogether. Then for parents with wrinkle and green, after segregation of gene occur, it will have a single and small r and small y. Then fertilization will occur. That is, the combination of this gamete will happen, which will produce the F1 generation, and all the F1 generation would give a capital R and a small r, a capital Y and a small y. That is, all the offspring are round and yellow. The reason why all the offspring are round and yellow, if you check, R is a dominant trait, right? And the small r is a recessive trait. The capital Y is a dominant trait, and the small y is a recessive trait. And we said that dominant trait, these are traits that express their effect in the presence of dominant trait. That is, the height or marks the dominant, the height or marks the recessive ones. Are we together? Now the next thing that will happen is now the crossing of the F1 generation. My board is filled up. Let me erase it, then I explain further. Now is the crossing of the F1 generation, that is, which is a capital R, small r, capital R, small r, a capital Y and a small y, right? Which I said they are round and yellow, heterozygous, round and yellow, heterozygous, round and yellow. Yeah, from your slide there is a mistake here. There's a mistake here. It ought to be heterozygous, round and yellow, not yellow and green. It's a mistake. All right. Now the next thing I said will happen is that there will be the crossing, right? That is the crossing of the F1 generation. The crossing of the F1 generation will happen, right? Crossing of the F1 offspring of the F1 generation. You know, the offspring of the F1 generation will be like this, a capital R, yeah. Now crossing of the F1 generation. Now this is where something new happens. Are we together? Yeah, this is where something new happens. Each F1 individual, that is, each F1 parent, that is, this is one parent, right? R and small r, right? Capital Y and small y, right? Now doing gamete formation in the F1 generation, that is, during gamete formation, the capital R separates from the small r. The capital Y, that is the yellow, it separates from the green, which is the small y. then they combine differently. Then they combine differently. They combine differently to form you have the RY, the R small letter y. This is R capital Y. I will explain. R capital Y, R small y, small r capital Y, small r small y. That is, they separate. That is, there will be a segregation of gene. Because I said that the Mendel first law of inheritance is very, very important to avoid inheriting double allele, which will lead to chromosomal abnormality. Are we together? That's why I said that during gamete formation for the second generation, that is for the F1 offspring, for them to combine, they ought to separate. Altogether, the round will separate from the wrinkle. The capital Y will separate from the small y, like this. Look at it. They will separate, right? They will separate to form something like this. Capital Y, capital R, and capital Y, capital R and a small y, right? Then the R, small r, and the capital Y, then the small r and the small y. Are we together? That is, before the parent, initially in the F1 generation, the parent had no variation. If you check, they had no variation. So each parent produced only one gamete. If you check in the initial write-up, you get to find out that each parent, look at it here, each parent produced just one gamete because they had no variation. But now the F1 generation, there is variation, so it produced different gametes. Are we together? It produced different gametes. That is, each parent now, after undergoing segregation of gene, they will produce 4 different gametes. Are we together? Before fertilization. That is, this one is for one of the gametes. Then this other one will also do the same thing. Are we together? You have they will segregate. You have something like this. You have something similar like this. Something like this also. We're together, right? Now, for you to determine the offspring of the F1 generation, you have to use the Punnett square. Yes, you have to use the Punnett square for you to be able to determine the genotype and the phenotype of the F2 generation. Now let's use the Punnett square to determine our next generation, that is the F2 generation. Let me wipe my board. Now, using the Punnett square to determine the offspring of the F1 generation, from our outline there, you see the gamete produced by the parent. This is the gamete of one parent. This is also the gamete of another parent. All right. Now, doing fertilization, the round and yellow combine with round and yellow to form homozygote round and homozygote yellow, right? I mean, the seed is round and yellow, homozygous. Then the RY combine with the capital letter R and small y to produce homozygous round and a heterozygous yellow, which is capital Y and small y. Then, which is capital Y and small y. Then the capital R and small y combine with small r and capital Y, that is wrinkle and yellow, to produce a heterozygous round and a homozygous yellow, that is a capital R and a small r, a capital Y and a capital Y. allele, which is heterozygous round and homozygous yellow. Then the RY combine with the small r and small y to produce a capital R and a small r, which is heterozygous round and heterozygous yellow. Together, alright. Then also this R, the capital R and the small y, it combine with the R and the capital Y to produce homozygous round and heterozygous yellow. The R combine with the capital R and small y to produce homozygous round and homozygous green. Yeah, this is small y, which is homozygous green. The RY combine with the small r and the capital Y to produce the capital R and small r, that is heterozygous round and heterozygous yellow. Then the R combine with this R over here, RY, to produce the capital R and small y, that is heterozygous round and homozygous yellow, small y, small y. We are together to this point. Then you pick the next one, the next garment. It combine the small r and the capital R combined with the capital R and the capital Y to produce the capital R and small r, that is heterozygous round and homozygous yellow. These combine with this to produce heterozygous round and heterozygous yellow. These combine with this to produce heterozygous wrinkle and homozygous yellow. These combine with this to produce homozygous wrinkle and heterozygous yellow. This is wrinkle and this is heterozygous yellow. Then these combine with this to produce heterozygous round, which is capital R and small r, capital Y and small y. We're together, right? Then these combine with this to produce a capital R and small r, that is heterozygous round and homozygous yellow. These combine with this to produce homozygous wrinkle and heterozygous yellow. Then these combine with this to produce homozygous wrinkle and homozygous green. That is, this is the offspring of the F1 generation. So when you count the results, you will have 9 round. Are we together? Let me turn it to my pen. You have 9 round yellow. 9 round yellow. After counting 1, 2, 3, 4, 5, 6, 7, 8, 9. We have 9 round yellow. We have 3 round green. 3 round green. 1, 2, 3. Then we have 3 wrinkle yellow. 3 wrinkle yellow. 1, 2, and 3. And also we have 1 wrinkle green. 1 wrinkle green. 1 wrinkle green. Look at it, a 1 wrinkle green. So what is happening here is that is what is happening from beginning to end is that the first cross, all the offspring, they are the same. If you take the first cross, that is, which is R, like the first cross, all the offspring, they are the same. But the second cross, many different combinations appear. And the reason is that because allele, they separate and mix in different way during garment formation. Are we together? During garment formation. That is what really happened here. That is the inheritance of one gene, that is the gene responsible for the shape, for the seed shape does not affect the inheritance of the seed color. That is what the second law, Mendel law, is talking about. That is the law of independent assortment of gene. The law of independent assortment of gene. And now, if you want to calculate the genotypic ratio, it will be the genotype will be 9:3:1. 9:3 ratio. No. The phenotype, it will be 9:3:1. That is 9 round yellow, 9 round and yellow, 3 round and green, 3 wrinkle and yellow, and 1 wrinkle and green. That is the phenotype, the physical appearance. The phenotype, it will be 9:3:1. Then the genotype, the phenotypic ratio, that is the genotype, the genotype now will be, now let's calculate capital R, capital Y. How many capital R, capital Y do we have? We have only 1. Then capital R, capital Y, and small y. How many do we have? Double R, 1. Look at 1, and 2. There are 2. Then the gene for R, capital R, then small y, we have 1. Let's check. Look at it. Capital R, small y, do double R, look at it, 1. That is round and green. Round, homozygous round and green, which is 1. Then heterozygous round and homozygous yellow. How many do we have? Heterozygous round and homozygous yellow. Homozygous yellow, 1. We have 1. This is 1, 1, 1, 1, and 2. We have 2. Then heterozygous round and heterozygous yellow, we have 4. Let's count together. Heterozygous round and heterozygous yellow. Heterozygous round and heterozygous yellow, 1, 2, 3, and 4. Then R, small r, and the small y. That is heterozygous round and homozygous green. Heterozygous round and homozygous green, we have 1. We have 1, 2. There are 2. Then homozygous wrinkle and heterozygous yellow. Homozygous wrinkle and heterozygous yellow, we have 1. That is only 1. Then homozygous wrinkle and heterozygous yellow, we have homozygous wrinkle and heterozygous yellow, we have 1 and 2. They are 2. Then the heterozygous wrinkle and the homozygous wrinkle, and the homozygous green, we have homozygous wrinkle and homozygous green, we have 1. So therefore the genotypic ratio would be 1 ratio 2 ratio 1 ratio 2 ratio 4 ratio 2 ratio 1 ratio 2 ratio 1. You know, in our previous video I distinguished between the genotype and the phenotype. I said that the genotype, it determines that the genetic makeup that determines a trait, it determines the phenotype, while the phenotype refers to the sum of all the observable feature of an organism, that the physical appearance, right? Then the phenotype of the F2 generation, the phenotypic ratio is 9:3:1. That is what you will see in the physical appearance of the offspring. It will be round yellow, round green, and a wrinkle yellow and one wrinkle green. We are together to this point. All right, now let's move to our next slide. Now the third law, that is the third Mendel law of inheritance, which is the law of dominance. This law explains why some traits appear while others do not appear. That is the law of dominance. That is Mendel's third law of inheritance. It explains why some traits appears while others don't. Because it says that according to this law, it states that a dominant allele hides or marks a recessive allele. According to this law, it states that the dominant allele hides a recessive allele. Hides the recessive allele. The recessive allele we only show, I explained this in the previous video, the recessive allele we only show when they are in two pairs. That is, when they are two recessive allele altogether, it only shows when they are two recessive allele. No doubt the law states that the recessive allele will always be marked by dominant allele. Yes, that is, the effect of the recessive allele will not be seen in the presence of the dominant allele altogether. So in essence, the Mendel third law of inheritance, that is the law of dominance, this law states that when two different alleles for a trait are present, the dominant allele will hide or mark the recessive allele. That is, the dominant allele will appear in the organism while the recessive traits will not be seen altogether. Now let me use this simple example to explain what I've been saying. Let's use seed color. Let me use seed color to explain what I've been saying. Seed color, right? Let's say that Y is the dominant, which represents yellow, is the dominant trait. Then the small y, which is green, let's say is the recessive, right? Let's say it's the recessive. Then the genotype YY, right, which is yellow, and Y and Y, which is still yellow, that is after crossing altogether. That is after crossing. That is, if a Y, a capital Y and a capital Y cross, it will still give yellow. That is what this law is talking about, because both of them, they are still dominant. If a capital Y and a small y, that is a yellow and a green cross, it will give yellow and green altogether, but the phenotype, it will still show yellow. Are we together? But the genotype, it will show that it is heterozygous yellow, that it is not a pure yellow. Are we together? Then the only way that the green will show its effect, that is, its effect will be seen when two recessive are present. Are we together? YY, which will give you a recessive green, which is a recessive allele, right? Which will give green color. The green color will only show its effect when two recessive traits they are present. Are we together? And also another important thing I want you guys to note here about the recessive trait is that even though this green is not showing here, right? Even though this green is not showing, despite it is not showing in the phenotype, it is still there. Are we together? It is still there, and that is, it is hidden and can appear in the next generation. That is, it is not lost. Are we together? Although the green allele is hidden, but is still present and can be passed to the next generation. Are we together? Yes, that is what the law of independence, that is what the law of dominance, that is the Mendel third law of dominance, is talking about. So in essence, the law of inheritance, that is the first law, the law of segregation of gene, the law of independent assortment of gene, which is the second law, and the law of dominance. This law explains how traits are passed from parents to offspring. Are we together? How traits are passed from parents to offspring. That is what this law we are all talking about. Now we'll be going into something very important, that is how this principle are applied in real life. How the principle are applied in real life, and this brings us to the history of medical and molecular genetics. Are we together? It brings us to the history of medical and molecular genetics, how these principles are applied in real life. All right, the history of medical and molecular genetics. All right, now what is medical genetics? Medical genetics, this is the branch of medicine that study how genes affect human health. It is the study of how genes affect human health and disease. That is medical genetics. It's a branch of medicine that study how genes affect human health and disease. Remember that Mendel talk about traits, right? Yeah. Mendel talk about traits. Right? In human, those traits can be sometimes referred to as disease. Are we together? Yes, you know that when we discuss extensively on the Mendel's law, that is starting from the history of Mendel, he spoke about traits, right? Then in human, those traits can sometimes be disease. Yeah, it can sometimes be diseases. So instead talking about, in Mendel law we talk about P color. Now in medical genetics we'll be talking about conditions like blood disease. Yes, in Mendel we spoke about traits, where we talk about the P color and the shape. But here in medical genetics we'll be talking about blood disease, genetic disorder. As I said, that the medical genetics is a branch of medicine that study how genes affect human health and disease, right? Here we'll be talking about blood disease, genetic disorder, inherited conditions. So in essence, what I'm saying here is that the Mendel law gave us the foundation. and medical genetics applies those law, right? Are we together? Yes. If you remember previously in our previous video where I said that the Mendel's work formed the foundation of modern genetics, right? Yes, the Mendel law or Mendel work, the Mendel law, it gives us the foundation. It gives us the foundation why medical genetics applies those laws. Yeah, medical genetics applies those laws for we to understand how disease, for we to understand disease process, how together to understand disease process, starting from the causation down to the progression of the disease, and also it also helps us to diagnose. Yes, medical genetics also helps us to diagnose them, that is to diagnose the disease, and also after diagnosis it also helps us to treat them altogether. So in essence, medical genetics is a branch of medicine that study how genes influence human health and diseases. Yes, it is a branch of medicine that study how genes affect or influence human health and diseases. Are we together? And also one key thing about this medical genetics is that it helps scientists to understand genetic disorder. Right? It helps scientists to understand genetic disorder. Yes. Genetic disorders, it occur. Let me wipe my bowl so I will explain properly. I said that medical genetics, it helps scientists to understand. Yeah, it helps scientists to understand genetic disorder. Yes. It helps scientists to understand genetic disorder and their causes. Genetic disorder, they occur when there is a mutation. Altogether, yeah, genetic disorder, they occur when there is a mutation, or mutation means when there is a problem, right? When there is a mutation or problem in a gene, when there is a mutation or problem in a gene or chromosome. Medical genetics helps scientists to understand genetic disorder and their causes. And I said that genetic disorder, it occurs when there is mutation or problem in a gene or chromosome. Like for example, taking sickle cell anemia, which is a genetic disorder, right? Yeah, sickle cell anemia. Sickle cell anemia is normally caused when there is a mutation in the gene that produce hemoglobin, right? Yeah, it is caused when there is a mutation in the gene that produce hemoglobin. Are we together? And this results to sickle cell anemia, because I said that sickle cell anemia is caused when there's a mutation in the gene that produce hemoglobin, and this hemoglobin is a very, very important protein. Yes, it's a very, very important protein in the red blood cell, and its function, that is the hemoglobin, right? Its function is to transport oxygen. Are we together? Its function is to transport oxygen to the body tissue. Transport oxygen to the body tissue in an organism, and if there is mutation in the gene responsible for the production of this hemoglobin, so therefore there will be inadequate supply of oxygen to the body. Altogether, yes, there will be inadequate supply of oxygen to the body, leading to hypoxia. That is, there will not be enough oxygen circulation in the body tissue. Altogether, and this sickle cell anemia from the word sickle cell, right? The red blood cell is sickle in shape. Altogether, since the red blood cell is sickle in shape, so therefore the hemoglobin inside, the hemoglobin is inside the red blood cell, right? Is also sickle in shape, and I said that the function of this hemoglobin is that it transports oxygen to the body tissue, and since it is sickle in shape, it will not be able to transport adequate oxygen to the body tissue. Altogether, no doubt people with the sickle cell anemia, they do come down with crisis. Yeah, they do come down with crisis due to inadequate oxygen supply to the body. And since there is inadequate oxygen supply to the body, there is a very high tendency of hypoxia. That is, there will not be enough oxygen. There will not be enough oxygen circulation to the body. And also, since there is no enough oxygen circulation, there's every tendency it might lead to necrosis. Yes, it might lead to necrosis. That's why you see patients with sickle cell, they do come down with joint problem. They are prone to infection. They have difficulty in breathing since there is no adequate oxygen supply to the body. Yeah, they normally come down with difficulty in breathing, joint problem, necrosis. They are also prone to infection. Yes, they are also prone to infection. We're together. I said that medical genetics, it helps scientists to understand genetic disorder and their causes. And I said that genetic disorder occur when there is a problem or mutation in a gene or chromosome. Then we take, for example, sickle cell anemia, which is a genetic disorder that is caused as a result of mutation in the gene responsible for the production of hemoglobin. Altogether, so another example of another genetic disorder is the Down syndrome. And Down syndrome is a genetic disorder that is caused as a result of an extra chromosome. Yes, an extra chromosome. That is, the organism inherits an extra chromosome. Yes, and also another example. When we go deeper, I will explain Down syndrome in detail. Yes, when we come to chromosomal abnormality. But now we are just laying foundation, right? Yes, I will explain deeper. Then we have other example of genetic disorder like the likes of the Phenylketonuria, which is caused by, let me write it, Phenylketonuria, which is caused by inability of the body to break down specific amino acid. Yes, the inability of the body to break down specific amino acid. And also secondly, medical genetics, they help scientists to understand how disease are inherited. Altogether, they also help scientists to understand how disease are inherited. You know that genetic disease are passed from parents to children through gene in the garment. That is through the sperm and the egg, which are the germ cell, right? Yeah, which are the garment. For male, the sperm is the garment, while for female is egg. Medical genetics. Let me wipe my board. Sorry. I said that medical genetics also help scientists to understand how diseases are inherited. That is, how these diseases are inherited. How these diseases are inherited. How these diseases are inherited. How the diseases are inherited. And you know that genetic disorder or disease, they can be passed from parents. The reason why that is, they are inheritable character since they are present in the garment, right? So therefore they can be passed from parents. They can be passed from parents to children. That is, from parents to their children through the garment, since these traits, they are present in the garment, and so therefore they can be passed. An example of such disease include the sickle cell anemia that we just spoke about, which is a genetic disorder that is caused due to mutation in the gene responsible for the production of hemoglobin, and also this sickle cell anemia is a genetic disorder that can be passed from parent to offspring. That's why I said that medical genetics help scientists to understand how diseases are inherited. I started by saying that medical genetics, it helps scientists to understand genetic disorder and their causes. Then secondly, it helps scientists to understand how disease are transferred from parent to offspring. An example of disease that can be transferred from parent to offspring include the sickle cell anemia, which is a genetic disorder. Yes, which is a genetic disorder. For example, the way this sickle cell anemia is being transmitted, if both parents are together, if both parents carry the sickle cell gene, that is, let's say the father have AS, the mother have AX, are together, yes, if both parents carry the sickle cell gene, a child can inherit the disease. Yes, and if only one parent carry the gene, the child may become carrier but show no disease. Are we together? That is, if the both parents carry the sickle cell gene, there's every tendency that the parents might give birth to sickle cell patient. Yes, there's every tendency the parent might give birth to sickle cell. That is, the offspring might have the tendency of showing the sign of the disease. Let's say this is the one parent, another parent, right? AS, they are carrier of sickle cell, right? Definitely, one after fertilization, they will give birth to AA. They will give birth to a carrier. Altogether, they will give birth to a carrier. Definitely, they must give birth to sickle cell individual. Despite this individual, at this initial stage, they might not show the sign of the disease, but they are carrier. Altogether, that is, if only one of the parents carry the gene, the child may become a carrier. But if both parents carry the gene, let's say both of them are SS, definitely they must give birth to children with sickle cell. Are we together? That is why medical genetics, it's very, very important. It's very, very important. Are we together? And also, medical genetics also helps scientists to diagnose genetic disorder. Yes, they also help scientists. They help scientists to diagnose. They help scientists to diagnose genetic disorders. They help scientists to diagnose genetic disorders. That is, doctors, they use different methods to detect genetic disorder. Like, for example, there's what we call the genetic testing. Yes, there's what we call the genetic testing. Let me erase the board. There's what we call the genetic testing, which is normally used in testing DNA. Yes, it's normally used in DNA testing to find if there is any mutation in the DNA. Are we together? I said we have the genetic testing. The genetic testing, which is normally used in testing DNA to check if there is mutation in the DNA, and also there is also blood test. Yes, blood test. I said that medical genetics helps scientists to diagnose genetic disorder, right? Like the example, the doctors use genetic testing to test if there is mutation in the DNA, and also there is blood test which is mostly used to detect disorder like the likes of the sickle cell anemia, and also there is prenatal testing, that is test that is being done during pregnancy. Prenatal testing, that is test that is being done during pregnancy to detect disorders like the likes of the Down syndrome. Is mostly detected during pregnancy, during prenatal testing. So medical genetics, it's very, very important. It helps scientists to understand genetic disorder and their causes, how diseases are inherited. It also helps scientists to diagnose genetic disorder. And also, one key thing about the study of medical genetics is that it helps scientists in adaptive measure in prevention. Yes, that is one of the key thing about medical genetics. It helps in prevention and treatment. It helps in prevention and treatment of genetic disorders or diseases. Yes, that is very key feature about medical genetics. After helping scientists to understand genetic disorder and their cause, how the disease are inherited, and how they can be diagnosed, then one key thing about the medical genetics, it helps scientists to adapt measure in prevention and treatment of genetic diseases. Yes, like through the use of genetic counseling, because medical genetics, they helps in preventing and treating genetic disorder, like the use of medical counseling. Medical counseling, let me write it down. Medical counseling, use of medical counseling, where they advise couples about genetic risks before having children. Yeah, like for patient, for patient or parents or couple or intending couples that they are carrier. Medical counseling will provide the knowledge that this couple they need about the genetic risk before having children, which will help them to curtail the risk of having sickle cell children, yes, later in life. Yes, thereby helping them to minimize the cost of living, because you know that medical treatment is very very expensive. Yes, it's very very expensive. A lot of resources do go with it, right? The genetic counseling, it will help advise couple about genetic risk before having children. Then in the prevention, there's also what we call in the treatment. Well, there's also what we call gene therapy in the preventive and the treatment of genetic disorder. There's what we call gene therapy, which involve replacing or correcting faulty gene, which is also one important of medical genetics. That is, in the treatment of genetic disorder, they use what we call gene therapy, in which faulty or in which faulty gene are being replaced. Then there's also in the treatment measure, there's also what we call special diet. For example, like patients with phenylketonuria, right? Phenylketonuria, that is in with the body, that is the inability of the body to digest certain protein, right? So such patients with phenylketonuria must avoid some certain food, right? Yeah, which is a preventive measure in order to avoid genetic disorder. They must avoid certain food in order to prevent complication. So in exchange, medical genetics study how gene affect woman and disease. That is how medical genetics affect human health and how it causes disease. It helps doctors to understand genetic disorder, right? How they are being inherited and how to diagnose them, and also how to prevent or treat them. That is what medical genetics is talking about. Are we together? That is, it helps doctors to understand the genetic disorder and their causes, how they have been inherited, and also how they can diagnose them, and also how they can prevent or treat them when they occur. All right, now let's move to the next thing we have in your slide, which is early discovery in medical genetics. So let me wipe my board, then we continue. All right, early discovery in medical genetics. From your outline here, number one, we have the phenylketonuria. Yeah, the phenylketonuria in 1934, right? In 1934. In 1934, a scientist called the Dr. Foiling discovered a condition called the phenylketonuria, which is also called as the PKU, right? Phenylketonuria. Now, what exactly is this PKU, right? The PKU is a genetic disorder in babies. Are we together? Yeah, it's a genetic disorder in babies. It's a genetic disorder in babies where the body cannot properly break down amino acid. Are we together? It's a genetic disorder in babies where the body cannot properly break down amino acid called the phenylalanine. Right together, the name of the amino acid that the body cannot break down is called the phenylalanine. That is where you get its name from, phenylketonuria, right? Phenyl and ketonuria. Phenyl amino acid ketonuria in urine, right? I'll let you go gradually. I said that it is a genetic disorder in babies where the body cannot break down an amino acid called phenylalanine. Normally, you know that when we eat protein, right, phenylalanine is being broken down by an enzyme, right? You know, when we eat protein, this phenylalanine is being, in normal individual, is being broken down by an enzyme, right? But in PKU patient, that is patient with phenylketonuria, that enzyme is missing. That is, the enzyme that is responsible for breaking this phenylalanine is missing or is not working. Are we together? I said that normally when we eat protein, phenylalanine is being broken down by an enzyme, right? But in patient with PKU, the enzyme that break down phenylalanine is missing or not working. Are we together? Then what happens is that the phenylalanine starts to accumulate. Since this phenylalanine is not broken down, are we together? Since it's not broken down, that is, since the amino acid is not being broken down, therefore it will begin to accumulate in the body, right? It begins to accumulate in the body. Then it becomes toxic. Are we together? Then it becomes toxic. That is something harmful, right? It becomes toxic, especially to the brain. We are going gradually, right? It becomes toxic, especially to the what? To the brain. Especially to the brain. It becomes toxic, especially to the brain. This can lead to intellectual disability if not treated early. Are we together? I said that the phenylketonuria is a genetic disorder, that is the PKU, in babies where the body cannot properly break down an amino acid called phenylalanine, right? And I said that when we eat protein, phenylalanine ought to be broken down by an enzyme, but in patients with PKU, that enzyme is missing, that it is not working properly. Then what happens is that since the phenylalanine is not being broken down, right, it begins to accumulate in the body, right? Then it becomes toxic, especially in the brain, to the brain, and this can lead to intellectual disability if not treated early. All together. And also the reason why it is called ketonuria is that I know, as I've explained the word phenyl, right, it comes from the word phenylalanine, right? Now let me explain the ketonuria. The reason why it is called ketonuria, maybe you may ask, is that from the word keto, right? Keto, from ketone body. You guys did chemistry, organic chemistry. From ketone, that is where you have keto from. Then urea, from urine, right? Because when phenylalanine builds up, are we together? When it accumulates, it is then converted. This phenylalanine is then converted to phenylketone. That is when it accumulates in the body. When phenylalanine accumulates in the body, it's then converted to phenylketone. That is the accumulation of phenylalanine as a result of not being broken down by the enzyme that ought to break it down, right? It then accumulates into what we call the phenylketone. Are we together? And this phenylketone is then passed out in the urine. That is why it's called phenylketonuria. Are we together? Yeah, it's being passed in the urine. That is why it is called phenylketonuria. Now let's link this to Mendel's law, because we said initially that the medical molecular genetics is just like the application, right, of the Mendel law. Now let's link it to Mendel law. Now PKU, right? PKU is a recessive disorder. We want to link it to the Mendel law because we said that the medical genetics is the application of the Mendel law, right? PKU is a recessive disorder, right? You know what is a recessive allele, right? An allele that only shows its effect in the presence of two recessive alleles, right? Let's link it to Mendel law. PKU is a recessive disorder. That means the child must inherit two of this defective allele for it to be present in the child. Are we together? That is, for this PKU to be present in a child, he ought to inherit one of the defective allele from the father, since it is a recessive disorder, and also one of the recessive allele from the mother. Are we together? Yes. Because we learned that recessive allele or recessive trait, they only shows their effects when the allele they are in pair, right? When they are in copy. This is how we connect the Phenylketonuria back to Mendel law. We're together to this point. All right. Now the last thing we have in our outline is the Klinefelter syndrome. The Klinefelter syndrome, it was discovered by Klinefelter in 1942. Yes, in 1942, which is an abnormality affecting mostly male. Are we together? It affects mostly male. It is as a result of an extra sex chromosome. Are we together? Yes, due to an extra sex chromosome. And this discovery led to further exploration in sex chromosome. This is what the second discovery by Klinefelter syndrome by Klinefelter in the year 1942. He discovered this is what we call the Down syndrome, right? Yes, the Down syndrome is what we call Down syndrome due to there's an extra sex chromosome leading to abnormality. The next thing from our outline here is the sickle cell anemia. All right, the sickle cell anemia, it was discovered by Dr. Herrick, right? It is a genetic disorder. All together, it is a genetic blood disorder. Let me wipe my board so that we explain this properly. Yeah, I said that the sickle cell anemia, which was discovered by Dr. Herrick in 1949. I said that the sickle cell anemia, it is a genetic blood disorder. The genetic blood disorder that affect the blood cells, most especially the red blood cell, that affect the blood cell. Are we together? Yeah, that affect the blood cell. You know that normally the red blood cell, that is in normal condition, I say it affect the blood cell, mostly the red blood cell. Normally the red blood cell is round. That is round in shape and it is flexible. Altogether, normally the red blood cell is round in shape and is flexible, and they carry oxygen using a protein called hemoglobin. Right. Then what causes the sickle cell is that in sickle cell anemia, I've explained this before. Let me just explain for the sake of what we have in here. I said that in sickle cell anemia, there is a mutation in the gene that produces hemoglobin. And this gene, hemoglobin, right? Mutation in the gene that produces hemoglobin. I've explained this previously, that produces hemoglobin. And I said that this hemoglobin is a protein, right, that is present in the red blood cell. All together, yes. And since there is mutation in the gene that is responsible for the production of hemoglobin due to an abnormality, right? There will not be adequate supply of oxygen in the body cell. All together, in sickle cell anemia patient, the red blood cell is sickle in shape. We are together, right? Because since there is mutation, all together, since there is mutation, that is small change in the DNA, Altogether, since there is small change in the DNA, the hemoglobin become abnormal. Altogether, that is, instead of the hemoglobin being smooth and sticky, instead of the hemoglobin on a normal, that is in a normal red blood cell, the hemoglobin, it is smooth. Altogether, it is smooth normally in a normal red blood cell. But if there is mutation, altogether, if there is mutation, it becomes sticky. Yes, it becomes sticky and rigid. That is, the hemoglobin becomes sticky and rigid. Altogether, and also, if there is mutation in the gene responsible for the production of hemoglobin, it becomes sticky and rigid. Altogether, and also what happens to the red blood cell is that the red blood cell, from its round shape, it then converted to sickle shape. I don't know if you are getting me. I said in exchange, in sickle cell patient, right? From the word sickle, the shape of the red blood cell is sickle in shape. I said earlier on that the shape of the red blood cell is normally is round and flexible. In sickle cell patient, the shape of the red blood cell is sickle in shape, and since it is sickle in shape, the hemoglobin also will follow the same shape of the red blood cell since it is inside. Altogether, and also there is also this thing. There is also a mutation in the gene responsible for the production of hemoglobin. So instead of hemoglobin, which is the protein that transports oxygen, instead of it to be smooth, it becomes sticky and rigid, and so therefore it cannot carry adequate oxygen to the body tissue. We are together, right? We are together to this point, right? So in exchange, the sickle cell of the red blood cell leads to anemia. That is why it is called sickle cell anemia. That is, the sickle cell causes anemia. And what is anemia? That is lack of blood. That is low red blood cell. That is the word anemia. Low red blood cell. Altogether, no red blood cell. And you know that the red blood cell, it normally breaks down after 120 days altogether. But an abnormal cell, that is in sickle cell patient, it breaks down very, very quickly. That is one key thing I want you guys to know. In sickle cell patient, the red blood cell, that is the abnormal cell, the abnormal sickle cell, it breaks down very quickly. It breaks down very quickly altogether, and since it breaks down very quickly, that it does not complete its normal lifespan. Since it does not complete its normal lifespan of 120 days, the body cannot replace them fast enough. Altogether, since the body cannot replace them fast enough, it results to what we call anemia. It results to what we call anemia, which is low oxygen supply to the body. That's what I'm trying to explain in exchange. Altogether, that is low oxygen supply. We are together to this point. Now let me recap then before we move further. Let me just recap a little. I said that in sickle cell anemia, it was discovered by Dr. Herrick in 1949, right? In 1949, he described the sickle cell anemia, and it said that the sickle cell anemia is a genetic blood disorder that affects the red blood cell. And I said that normally the red blood cell are round. They are round and they are flexible in shape. They are round and flexible, right? And they carry oxygen using a protein called hemoglobin, right? Then in sickle cell patient, there is a mutation in the gene that produces hemoglobin. And is this gene the gene responsible for producing hemoglobin inside the red blood cell. That is, there is a mutation. That is, there is an abnormality in that gene. Are we together? And the word mutation simply means when there is a small change in the DNA. That is what mutation abnormality. Are we together? Then the hemoglobin become abnormal since there is mutation in the gene responsible for production or for producing hemoglobin. Are we together? That instead of the hemoglobin to be smooth, it becomes sticky and rigid. Right? So now we move to what happened to the red blood cell. In sickle cell patient, the red blood cell, which is normally round, becomes sickle in shape. Are we together? And this sickle cell shape of the red blood cell is rigid. Are we together? And it is destroyed early, because you know that I said that the normal lifespan of the red blood cell is 120 days before it's been destroyed then produced back, right? In sickle cell patient, it's been destroyed very fast, and since it's been destroyed very fast, and since it does not follow the normal lifespan of the red blood cell, that the body will not be able to replace them very fast. Out together, since it does not complete its normal 120 days lifespan, right? Then the body will not be able to replace them very fast, which results to anemia, which is low oxygen supply to the body. Out together. Now let's connect this to make it interesting to Mendel law. That is what we've learned earlier, right? Then sickle cell anemia is also a recessive disorder. Yes, it's also a recessive disorder. It's also a recessive disorder. That is, XS has the disease, right? Has the disease. Then AS is a carrier. AS is a carrier. AA is normal. Now, so again, the disease only appear when a person has two recessive alleles. You know what is recessive allele, right? So now, what Mendel discovered in pea plant, now we are seeing it in real human disease. We are together. That is where I want to link it up. That is for it to appear in an individual, so that individual inherit the both allele from the parents. Are we together? So what we are seeing in pea plant now, we are seeing it in a real human disease. All right, now I will leave you with a question which you will drop on the comment section. All right, you drop it on the comment section. For instance, now, if both parents, right, if both parents are carriers, that is, if both parents are AS, right, can they have a child with sickle cell? I would like you to drop it on the comment section. Now let's move to what we have next on our slide: the emergence of molecular genetics. All right, let me wipe my board, then we'll continue. Okay, the emergence of molecular genetics. All right, when we talk about the emergence of molecular genetics, one of the most important breakthrough that happened in 1953, at this time, that is in the time of James Walton, James Walton. At the time of James Walton and Francis Crick, is that they discover the helix structure. Altogether, they discover the helix structure of DNA. The helix structure of DNA, and this discovery was very, very important because it helps scientists understand how genetics information is stored. Altogether, the important breakthrough that happened during the emergence of molecular genetics at that time, and that is during the time of James Walton and Francis Crick, is that they discovered the double helix structure of DNA. And this discovery was very, very, very important because it helps scientists understand how genetic information are stored, and how it is being passed from one generation to another, and how it is being passed from one generation, and how it is being passed from one generation to another. Altogether, that is the important breakthrough that happened in 1953. Then moving in 1953, 1953. That is during the time of Francis Crick and James Watson. They discovered the double helix structure of DNA, and this discovery was very, very important because it helps scientists understand how genetic information is being stored and how it is being passed from one generation to another. Then moving further in the 1970s, in the 1970s, another important or another major breakthrough occurred with the discovering of the restriction enzyme. Yeah, another important breakthrough occurred with the discovery of the restriction enzymes. These enzymes, they are special because they can cut DNA at specific points. Please get this thing. Another major breakthrough occur with the discovery of this restriction enzyme, because this restriction enzyme, they are special because they can cut DNA at specific points altogether, and this make it much easier for scientists to study gene and even manipulate DNA, leading to major advances in genetic research altogether. That is the major breakthrough that later occurred in the 1917 is the discovery of the restriction enzyme, and these enzymes, they are special because they can cut DNA at specific point, and this make it easier for scientists to study gene and even manipulate gene, since it can cut the restriction enzyme can cut gene at specific point altogether, and this discovery leads to major advances in genetic research. That is, in essence, this discovery helps scientists to understand DNA better altogether. That is the discovery of the restriction enzyme. It helps scientists to understand DNA since they can manipulate its DNA better altogether, and this opened the door to modern genetic technology altogether. That is what the emergence of molecular genetics, that is starting from the time during the time of Watson and Francis Crick, where they discovered the double helix structure of DNA, and this discovery helps scientists understand how genetic information is stored and how it is being passed from one generation to another. That is in 1953. Then moving further in the 1970s, another major breakthrough occurred with the discovery of the restriction enzyme. These enzymes, they are special because they can cut DNA at specific point, and this make the work of scientists to be much more easier for scientists to be able to study gene and even manipulate DNA, and it leads to major advances in genetic research. That is, it opened door to modern genetic technology. We are together to this point. Alright, now let's move to our next slide. Alright, the recent breakthrough in medical genetics. Okay, let's talk about some of the recent breakthrough in medical genetics. Let me just put it in a simple way. Firstly, in 2017, there was a major breakthrough in treating blindness. Altogether, there was a major breakthrough in treating genetic blindness. Scientists, they developed a medicine called the Luxturna. Altogether, they developed a medicine called the Luxturna. This treatment, it works by putting a healthy gene into a patient's eye, especially the retina. Altogether, I said that let me just put the recent breakthrough in genetics in a simple way for you guys to understand. I say firstly, in 2017, there was a major breakthrough in treating genetic blindness. That is associated with gene, right? Scientists, they developed a medicine called the Luxturna. This treatment works by putting a healthy gene into the patient's eye, especially the retina. Altogether, especially the retina of the patient's eye, they put a healthy gene, especially in the retina, a healthy gene into the patient's eye, especially the retina. So if someone is blind because of faulty gene, a healthy gene is added to help restore vision. All together, that is the major breakthrough that happened in 2017, in which scientists they treat genetic blindness by using a medicine called the Luxturna. It works by putting a healthy gene into a patient eye, especially the retina, right? Then, so in action, if someone have blindness as a result of faulty gene, a healthy gene is being added to help restore vision. So the next is in 2019. In 2019, scientists they use a powerful tool known as the CRISPR, which is called the cluster regular interspecies short palindromic repeats. Right? That is the CRISPR. Um, the CRISPR, these two allow scientists altogether. It allowed them to cut. Please get this thing. To cut and fix DNA. It allows scientists to cut and fix DNA, just like you are editing a document. Altogether, yes, it allows scientists to cut and fix DNA, just like you are editing a document. And also, it was used to correct gene responsible for sickle cell anemia. Altogether, yes, it was used. The CRISPR was used in correcting gene responsible for sickle cell anemia. Altogether, yes, this the CRISPR is just like a tool. It allows scientists to cut and fix DNA, like the way you edit your document. Altogether, so in simple terms, the CRISPR, they can edit bad gene. They can edit bad gene. Altogether, they can edit bad gene and replace them with correct ones. That is what this is talking about. I replace them with correct one. That is also another breakthrough that happened in medical genetics. So in excess, sickle cell anemia can now be treated. Altogether, it can now be treated using CRISPR, which is a tool that allows scientists to cut, edit, and fix DNA, like just the way you are editing a document. And it was used to correct gene responsible for sickle cell anemia, which is a disease that affects the red blood cell. Yes, it was used to fix it. All right. Now, another recent breakthrough that happened in the 20s, in the 2020s, the 2020, is the artificial intelligence, that is AI, right? Yeah, AI became very useful. Let me write it. AI became very useful in medicine. AI was used to analyze. It was used to analyze medical images. It was used to analyze medical images and also detect genetic disease easily. That is the recent breakthrough that happened in the 2020s. Artificial intelligence became very, very useful in medicine. AI was used to analyze medical image, that is in the MRI, CT scan, right? Yes, and also it helps to detect genetic diseases. That is, in exchange, computers can now help doctors find diseases faster and more accurately. Altogether, the likes of the MRI, the CT scan, yes, which are computers that helps in diagnosis. We are together to this point. These are the recent breakthrough in medical genetics from 2017 down to 2020. Are we together? So now we'll move to the next thing we have in our slide, which is cytogenetics. All right, let me write cytogenetics. Cytogenetics is a branch of genetics that study chromosomes in our cell. Altogether, It's a branch of genetics that studies chromosome. We study chromosome. We study chromosome in our cell. That is, it focuses on how many chromosomes are in a cell. Altogether, I said that cytogenetics, the branch of genetics that study chromosome in our cell, it focuses, let me clean the board and we'll continue. That is, it focuses on how many chromosomes. That is, cytogenetics, it focuses, I said that it deals with the study of chromosome in our body. It focuses on one, how many chromosomes do we have altogether. That is, how many chromosomes are there in a cell or a living thing, how many chromosomes are there in a living thing, and also how chromosomes look like, how chromosome looks like, that is their structure, right? And also it also focuses on what chromosome does in the body, that is their function, what they do in the body. And also another important thing is that it focuses on what happened when chromosome change. What happened when chromosome change? We are together to this point. What happened when chromosome changes? So in essence, cytogenetics is the study of chromosome. Altogether, it is the study of chromosome, how many they are, that is the number. Altogether, how they are arranged, that is how they look like, that is the structure, and how they are arranged, what they do, that is their function, and also what happens when something go wrong with them. We are together to this point, right? For example, to help you understand this properly, in women we have 46. That means the number, right? To answer the question, in woman we have 46 number of chromosomes altogether. That is, and they are 23 in pairs. Altogether, they are 23 in pairs, and but sometimes, right, a person may have an extra chromosome or a missing one altogether, and this can cause what we call genetic disorder. Like take for example, like patient with Down syndrome. Yes, patient with Down syndrome. What really happens here is that there is an extra copy of chromosome, that is the chromosome 21 altogether. So in essence, is a branch of genetics that study chromosome in our cell altogether, and it focuses on how many they are altogether, how many number of chromosomes an individual have or in a cell, and also how they look like, that is their structure and their function, and also what happens to them when they change. We are together. For example, like in an in a living organism, that is in woman, a woman ought to have 46 chromosomes, 23 pairs altogether. When the number exceeds 23 pairs, there is an abnormality. Like in an individual with Down syndrome, there is an extra chromosome altogether. Yes, there is an extra chromosome, that is chromosome 21, which is a genetic disorder altogether, leading to what we call Down syndrome. That is, if the number of chromosomes is not up to 46 or is more than 46, there is a genetic disorder altogether. So in essence, cytogenetics study of chromosome altogether. It look at the number, the structure, and function. Look at the number, structure, function, and abnormality. We're together, right? Now the next thing on our slide is DNA. What is DNA? Alright, let me wipe my board, then we kick off.